Hi, year 11s and 12s. Today we're looking at an IB PowerPoint. This is a HL content. We're looking at photosynthesis. This is the third PowerPoint and the third subtopic of metabolism, respiration, and photosynthesis, and the last lot of content we need to know before our topic test on topic 8. As I said, this is HL content. There's going to be a number of videos in this PowerPoint. So the first one was going to look at an overview of photosynthesis, separating it into light-dependent and light-independent reactions, talking about the structure of the chloroplast, for example. We're then going to talk specifically in another video about light-dependent reactions. Then we're going to have another video on light-independent reactions, and lastly we're going to have a video on the Calvin cycle. So, for now, let's begin starting to talk about photosynthesis. The first thing we need to do, so there won't be a need to know in this video. They are on the PowerPoints in the OneDrive if you'd like to look at that, but they're not in this video. So let's start by looking at the chloroplast structure and function. So, the structure and function of chloroplasts is adapted to help function in photosynthesis. So the chloroplast has basically adapted to help with photosynthesis. We know that chloroplasts are like solar energy plants. They convert light energy from the sun into chemical energy. And then that chemical energy can be turned into ATP through light-dependent reactions or organic compounds through light-independent reactions. Only photosynthetic tissue possesses chloroplasts. So, for example, it's in leaves, but we can't find it in roots because they don't have photosynthetic tissue in the roots of plants. We believe that chloroplasts have once been independent prokaryotes. That's endosymbiotic theory that we talked about in topic 1. But they were internalized by eukaryotes via endosymbiosis. They have a double membrane base because of the endosymbiosis they've gone through. They have their own DNA, which is circular and naked, so ribosome 70S. And their metabolic processes are susceptible to certain antibiotics. Now, you have to know the structure, so you have to be able to draw a chloroplast. You also have to be talking about the different parts that are there. So, for example, let's start with the thylakoids here. So the thylakoids are flattened discs. They have a small internal volume to maximize the hydrogen gradient upon proton accumulation. We also have the grana, or granum, as it's labelled in the diagram over here. Basically, these are thylakoids arranged into stacks, and we do this to increase the surface area volume ratio of thylakoid membranes. We have our photosystems, which are not actually visible in this diagram here. Basically, they're the pigments that are organised in thylakoid membranes to maximise light absorption, so they're inside. We also have our stroma, which we can see over here, which is the central cavity contains enzymes and a suitable pH for the Calvin cycle to occur. And lastly, we have a lamella or lamellae, which connects and separates thylakoid stacks. You can see this thin system here, which helps to maximise photosynthetic efficiency. So that's how you'd label a chloroplast if you had to. That's what the components do to help their function. Now you also need to be able to show diagrams of chloroplasts and their function relationship. So we've got electron microscopes and our diagrammatic representations here. So electron microscopes of chloroplasts are quite different to what we're physically drawing, depending on where they do the cross-section of the chloroplast. So typically they should show that they are round in appearance, so we can see there's almost round in appearance here, and you've got that double membrane that we can visibly see in the chloroplast there. You can see your flattened discs, thylakoids, arranged into their grana, okay, and you can see very thinly the lamella connecting them all together. Not much visible space inside there. In fact, I would generally suggest what we're seeing here is not ribosomes and chloroplast DNA. In fact, I'd suggest what you're seeing here is the starch granules, which appear as dark spots within the chloroplast. So these little dark spots are not ribosomes or circular DNA. Instead, what we're looking at here is starch granules. Okay, so that is how to compare an electron microscope diagram of a chloroplast with the actual diagrams we're physically drawing. So, let's talk about the two different reactions. We have light-dependent reactions and we have light-independent reactions. Light-dependent reactions for photosynthesis convert light energy from the sun into chemical energy, ATP, and our light-independent reactions use chemical energy to synthesize organic compounds such as carbohydrates. Okay. The first thing that happens is our light-dependent reactions, which is when light is absorbed by chlorophyll, which releases energized electrons that are used to produce ATP. Those electrons are donated to carrier molecules, NADP+, which are used along with ATP in light-independent reactions. The electrons that are lost from the chlorophyll are replaced by water, which is split through a process called photolysis to produce oxygen and hydrogen. The light-dependent reactions will then occur in the intermembrane space of the membranous discs called thylakoids. So the main thing we're taking from this is, one, that light-dependent reactions are the first step in photosynthesis, and as the understanding here is that they're actually physically happening in the intermembrane space of the thylakoids, whereas our light-independent reactions, which is our second step in photosynthesis, when the ATP and hydrogen electrons are carried by NADPH and are transferred to the site of light-independent reactions. The hydrogen, or the electrons, will be then combined with carbon dioxide to form complex organic compounds, like carbohydrates, such as glucose, and the ATP will provide the energy to power those anabolic reactions and fix those carbon molecules together. Those light-independent reactions will occur within the fluid-filled interior of the stroma. So again, our understanding here, that light-independent reactions are taking place in the stroma. And that is the end of our first video.